Anybody here love math? Nobody's a math lover? Well, today's math is not very scary. In fact, you do not even need to use a calculator, just your brain. So we'll see how well your brains are working today. So the experiment that we do is the serial dilutions and standard plate counts. There's also a part that we do that I'm not doing remotely, which involves using a spectrophotometer to measure the absorbance of a bacterial culture. I'll talk a little bit about it and we'll watch a video on how to use the spectrophotometer, but I don't actually do that remotely. So we're just going to focus on how to calculate serial dilutions and calculate the number of cells per milliliter in the original sample. So when it comes to a serial dilution, we're going to perform a series of dilutions. And by the end of the process, we'll be diluting our original bacterial sample by tens of millions of times, maybe hundreds of millions of times. So it's a very simple but very powerful technique. It does involve some math, but it's not complicated math. So when it comes to a dilution, when you calculate a dilution, it's always the part divided by the whole. The part is the volume that you add to the tube. The whole is the total volume now in that tube. So we typically work with 9 milliliter sterile broth or sterile water tubes to do the dilution. And we typically add 1 milliliter of our solution to be diluted into those 9 ml tubes. So the part that we add is 1. This is 1 ml. The total volume now in that tube is 9 plus 1, or 10 mLs. So 1 over 10, or a 1 to 10 dilution. So 1 over 10 is the same as 0.1, or 1/10. But we do like to use exponents because we need to use them for the calculation. So 1/10 is the same as 10 to the negative 1. And yes, technically it's 1 times 10 to the negative 1, but it's 1 times everything. And so we understand that, so we don't have to say that. We can just talk about 10 to whatever the exponent is. So far so good. Part divided by the whole. Right, the volume you add divided by the total volume now in that tube. Right, so that's pretty straightforward, yes? Add 1 ml into 9, that's a 1 to 10 dilution, or 0.1, or 10 to the negative 1, yes? Let me know you follow. So do we all follow that so far? Yes? You gotta give me some feedback. I feel like I'm not talking to anyone but myself here. Now, we can also use 99 milliliters of sterile water or broth to do a dilution. So if you add 1 ml into 99, that's 1 over 100. And 1 over 100 is 0.01 milliliters, or 0.01. And then exponentially, that's 10 to the negative 2. So you can, either use 9 ml or 99 ml, and we do that to keep things simple. But you can use different, quantities to do your dilutions. But most often we use 9 ml tubes or 99 ml bottles. Now, when it comes to dilutions, they are cumulative. So if you performed a 10 to the negative 1 dilution first, and then you performed another 10 to the negative 1 dilution, your total dilution would be 10 to the negative 2. If you did a 10 to the negative 1 dilution first, and then you did a 10 to the negative 2 dilution, so you added the 1 ml into 99 mLs, your total dilution would now be 10 to the negative 3. So the idea is that the dilutions are cumulative. So if you do repeated 10 to the negative 1 dilutions, as is the case here, right? You dilute the first one 10 to the negative 1, and you do another 10 to the negative 1 using the next dilution tube, and another 10 to the negative 1 doing the next dilution tube, and so on and so forth, and we do all these 10 to the negative 1 dilutions, our final dilution is 10 to the negative 8, because 10 to the negative 1 times 10 to the negative 1, etc., etc. You add the exponents, right? So our cumulative dilution would be 10 to the negative 8. So the key point here is that dilutions are cumulative. If you do a 10 to the negative 1 dilution and you do it again using that first 10 to the negative 1 dilution, your total dilution is going to be 10 to the negative 2. If you do another 10 to the negative 1 dilution on the tube that was diluted 10 to the negative 2, it becomes a 10 to the negative 3 dilution, and so on and so forth. So each time you're becoming more and more dilute. And the dilutions are cumulative, so you multiply them, and of course when you do that with exponents, you end up adding the exponents together, right?
So, Professor, in serial dilutions, right? You first add the part and the whole, then you divide parts with the whole, right?
Right.
But in cumulative, you just add the exponents.
Right, because it's like multiplying everything together, 10 to the this times 10 to the that, and so you add the this and that together.
Okay, thank you.
So like here we did our first dilution, and then if we take this tube and we dilute it again 10 to the negative 1, our total dilution is 10 to the negative 2. And if we take that and dilute it again to the negative 1, our total dilution becomes 10 to the negative 3. And then 10 to the negative 4, 5, 6, 7, 8 as our final dilution. So the dilutions are cumulative as you go. So that's all well and good. That allows us to calculate the dilution. But in order to do our final calculation, we need to use what's called the dilution factor. And the dilution factor is the number of times the original solution has been diluted. Mathematically, the dilution factor is 1 divided by the dilution. So 1 over the dilution. 1 over a negative exponent, and the exponent becomes positive, right? So if your dilution is 10 to the negative 3, 1 divided by 10 to the negative 3, it becomes 10 to the positive 3. So your exponents simply become positive, and that is your dilution factor. So in the experiment that we're doing, each group gets a sample of E. coli. And so the E. coli broth is very cloudy. It's very turbid. So you got millions upon millions of cells in there per milliliter. If you were to take a small drop of that and spread it on the surface of an agar plate, you get solid growth. And that doesn't tell you anything. What we want to try and figure out is what is the concentration of cells per milliliter in this original sample. And so the way we're going to do that is with a serial dilution, and then we're going to spread samples onto the surface of agar plates and hopefully obtain isolated colonies that we can count. So each group gets 6 9 ml dilution tubes. You take 1 milliliter out of your stock, you add it to the first tube, and since it's 1 into 9, that's a 1 to 10 dilution, is 10 to the negative 1. Then you mix this tube thoroughly and take 1 milliliter from this tube, add it to the next 9 ml sterile tube. So you're doing another 10 to the negative 1, but you're diluting a sample that's already diluted 10 to the negative 1. So negative 1 and negative 1 makes negative 2. Mix this tube thoroughly and then transfer 1 milliliter from this tube into the next one. So that's another 10 to the negative 1 dilution, but this tube was already 10 to the negative 2. So negative 1 and negative 2 makes negative 3. And then you repeat that, and you end up with a final dilution of 10 to the negative 6. So a negative 1 dilution, another negative 1, another negative 1, and so on and so forth, and your total is a 10 to the negative 6 dilution. And then what we're going to do is, starting with tube number 2, we're going to take samples from each tube and spread them on the surface of agar plates. Now, if you were taking a 1 milliliter sample and spreading that, you don't have to do any further calculation to your dilution. However, we're not taking 1 milliliter. We're taking 100 microliters. How many milliliters is 100 microliters? Remember, microliter is 10 to the negative 6. Milliliter is 10 to the negative 3. And so, yes, Maria. So what you would do is just simply move the exponent to the left 3 places, and that gives you 0.1 milliliters. Right? So 100 microliters is equivalent to 0.1 milliliters. Well, we already know 0.1 is 1/10. 1/10 is 10 to the negative 1. So if you multiply this, a 10 to the negative 1, times the dilution in the tube, you get the effective dilution on the surface of the plate, which is 10 to the negative 3. So if you are taking samples that are not 1 milliliter, you have to multiply the volume that you take times the dilution of that tube, and that gives you the effective dilution on the surface of the plate. So again, 10 to the negative 1 sample times 10 to the negative 3 gives you 10 to the negative 4. Negative 5, 6, 7, right? Now, in one of the videos that we see, they wait till the end to talk about the volume that they put on the plates. If you wait to the end of your calculation, you actually have to divide your number by that volume. If you do it early like this, we multiply it times the dilution. But if you wait, after you get your number at the end, you then divide it by the amount you put on the plate. But you'll see that in the video. So it's just 2 different ways to do it. You get the exact same number at the end. I like to do the dilutions up front so that I know what the dilution on the plate is. And that will simplify my calculation at the end. But that's only if you're using an amount that's not 1 milliliter. If it's 1 ml, then nothing changes, right? Because 1 times anything is the same thing. And anything divided by 1 is the same thing. So if you use 1 ml, you don't have to worry about doing any further manipulation of your numbers because it's 1 ml. But if it's 0.1 or anything else, then you have to factor that into your calculation. So we've calculated all of our dilutions. What we need is the dilution factor. And remember, that's 1 over your dilution. So 1 over a negative exponent becomes positive exponents. And there is a typo here. That should be 10 to the 5, not negative 5. So we simply make the exponents positive. So that's our dilution factor. So in terms of the number of times we've diluted our sample, in this first plate here, we've diluted the original bacterial sample by 1,000 times, right? 10 to the 3. Here is 10,000. Here, 100,000, 1 million. And by the end, we have diluted the original sample by 10 million times. That's a lot of dilution, right? But that's what we need to do in order to dilute the bacteria enough so that we get isolated colonies on the plate that we can count. So that's the power of a serial dilution. Using just 6 sterile 9 ml tubes, we've diluted the original sample by up to 10 million times. So, how do we do the final calculation? Well, what we do is we are going to multiply the number of colonies on the plate times the dilution factor of that plate. Now we're going to see some examples on the next page, but our range, right, that is acceptable for the number of colonies is between 30 and 300. If it's more than 300, we say it's too numerous to count, or TNTC. If it's below 30, we say it's too few to count, or TFTC. And the reason why that is, there's significant experimental error when you're pipetting. So in general, if the number is below 30 colonies, it's not considered to be accurate. When it comes to more than 300 colonies, those colonies tend to grow into each other, and so it's hard to see them as individual colonies. So again, it's less accurate if you're above 300. So that's what our range is going to be, 30 to 300. So here we have some sample data, right? So you were counting colonies. The first plate was covered with bacteria, it's TNTC. The second plate, again, covered with bacteria, TNTC. On the third plate, you were counting, but you continue to count above 300. So again, we cannot use this plate because it's above 300. Next plate was 47. The last plate was 5. So obviously 5 is too few to count. So this plate here is the one that falls between 30 and 300. So this is a plate that you would use. So again, in order to estimate the number of CFUs per milliliter, and a CFU is a colony forming unit, and we know that a colony is produced by a single cell. It's the same thing as cells per milliliter in most cases. So we take 47, multiply it by the dilution factor, and boom, right? How hard is that? 47 times 10 to the 7. But there is a problem with that number. What's the problem with that number? Why can't you write 47 times 10 to the 6?
Because both has to be in scientific
Notation, right. Yep. So we always have to write our numbers in scientific notation. So the proper way to write that is 4.7 times 10 to the 7, right? Because we make 47 smaller by 1 decimal, we increase the exponent by 1 decimal. And that way, it's the same number, right? So 4.7 times 10 to the 7 CFUs per milliliter. And we did all this without the help of a calculator, right? Because you don't need a calculator to do these things. So what does this number mean? 4.7 times 10 to the 7? That means we had 47 million cells in every milliliter of broth in the original culture, right? The one we started with. But of course, that was only at that particular point in time where we took the sample. An hour later, that number would be different. But we were estimating the number of live cells per milliliter in the original sample. And we can do that by using a serial dilution, and in this case, the spread plate technique, where we spread our samples on the surface. In one of the videos, you'll see the pour plate technique, where they put the sample in a sterile petri dish and then they add melted agar to it. Either way works. We just prefer to do the spread plate method rather than the pour plate because it's a lot easier to do. So, but that's how you do a serial dilution. In your head, no calculator required, right? So questions? Now's a good time for questions. Here we see some actual plates from the experiment. Again, the first three are TNTC. This plate here is between 30 and 300, and this one is just less than 30, so we don't use it. So only this one. So you find the plate that has colonies between 30 and 300, multiply that number of colonies times the dilution factor of that plate, and that gives you CFUs per milliliter. And just make sure to put the number into proper scientific notation. So in theory, only one plate should be between 30 and 300, right? So are you guys familiar with the T's test? What's the T's test? What's the T's test? What's T's? TEAS. Who takes it? Nobody knows T's? Right, so it's sort of the SAT for nursing students. And sometimes they have dilution problems on the T's test. So they might ask you something like, they'll give you these numbers and they'll give you this number here, and they'll ask you to estimate if there's 47 colonies on this plate, how many colonies would you expect to find on this plate? So you know this, how many colonies would you expect to find on this plate? Right, because they wouldn't give you this number. So how many colonies would you expect to find on that plate if you know that the 10 to the 6 plate has 47 colonies on it? What would be your estimate on the 10 to the 5 plate?
$45.
So why do you think 25? So remember, we're getting more dilute as we go, right? And so each of these numbers differs by a factor of 10. So if you know the 10 to the 6th plate is 47, how many colonies would you estimate to be on the 10 to the 5 plate? If there's 10 times difference between these, and this is 10 times fewer than this plate, what would be your estimate?
Professor, say it again, please. Say your question again.
So again, we're given this number. I'm asking you to estimate how many colonies would be on this plate based on this number. Each plate is 10 times more dilute than the plate that preceded it. So what would your estimate be for this plate? How many colonies? You guys are making this too hard. Sarah? Roxy? Bryant? Maria? Any ideas? There's going to be a lot of dead air on this recording. So what would you predict the number of colonies to be on the 10 to the 5 plate? If 10 to the 6 is 10 times more dilute. Well, yes. No, but how many? The 10 to the 5 plate. What would you predict the number of colonies to be based on the 10 to the 6 plate? And yes, it's going to be more. How many times more? No, that was the estimate that I gave in the example. But I'm asking you, if you don't have any numbers except for 47, make some predictions. What are your estimates going to be? You guys must have had a rough weekend. The 10 to the 5 plate has 10 times more than the 10 to the 6. So what would your estimate be? Right, so around 470, 500 would be your estimate, right? 10 times more. And so what would your estimate be for the 10 to the 7 plate, which has 10 times fewer cells on it than the 10 to the 6 plate? Well, somewhere between 4 and 5 if your technique was perfect, right? So it'd be 10 times fewer. And so the whole point is that if you know one of the numbers on the plates, then you can estimate what everything else should be, right, based on that number, right? So if we know there's 47 colonies here, this plate has 10 times more, it should be around 470, 500, just as a rough estimate. And this plate should be between 4 and 5, right? This would be 4,700, right? So way, way too many cells, but you should be able to make estimates, right, based on at least one of these numbers as to what you should get. And that's assuming that your technique is perfect, which sometimes the student groups, they get some excellent results where their numbers show excellent technique, right? They're exactly what you would predict, so their technique was really good. Now, one of the other things that we do in the face-to-face class, which we're not doing here, is that you would also take your original stock solution and you would measure the absorbance in a spectrophotometer. And so there is a lab that has created a calculator where you can enter the absorbance value for your culture, and it gives you an estimate of CFUs per milliliter based on that absorbance value. And so it's interesting to see, again, if the internet, the web estimate is very close to your actual results you get when you do the experiment. And if they are very close, then that means your technique was really good. If your calculated value is way off, then that means your technique was not very good. So it is interesting to use that for comparison. So we will talk about the spectrophotometer. There are some problems with using it to measure bacterial growth. The biggest problem is that live cells and dead cells absorb equally. So when you use a spectrophotometer, the absorbance value is giving you the value for both live and dead cells. We only want to know the number of live cells in a culture. And so the web calculator takes that into account when it gives you the estimate of CFUs per milliliter. And that is actually just using E. coli, because E. coli is a single bacterium. It doesn't stick to other E. coli cells, because if the bacteria stick together, that creates a problem. If you have a cluster of cells, the spectrophotometer cannot interpret the number of cells that are there in the cluster. Or if cells are in chains, long chains, a spectrophotometer is not going to be able to interpret those data, right, to give you an accurate estimate. So our web calculator actually was just designed to work with E. coli in the spectrophotometer. So it doesn't work for other kinds of bacteria. So there are some problems, right, with clustered bacteria. They don't work well for estimating in the spectrophotometer. And again, live and dead cells absorb equally. So those are some limitations of using a spectrophotometer. Our technique here just measures live cells. So questions? Now we're going to watch some videos. So I'll share my screen. I'll lower the volume a bit. So all these links are on Blackboard. And this is just how to use the spectrophotometers that we use at Rockville. So if you took Biology 150, you also use these in 150 at Rockville. And what we're actually going to see in the video, the student or the technician is actually measuring a chloroplast extract, which is what you did in 150. But we will just watch this just so you can see how the spectrophotometer is used.
In this video, I will demonstrate how to use the Genesis 20 spectrophotometer in biology lab. The spectrophotometer will be used to measure the absorbance of a variety of samples in the lab. The Genesis 20 spectrophotometer has three main parts. The display shows the wavelength that has been set and the absorbance value. The interface is the panel that contains the controls to set the wavelength, blank the machine, and set the type of reading. In our case, this is absorbance. The last part is the sample chamber where sample cuvettes are placed. Special sample cuvettes are needed to take absorbance readings. These are the two types of sample cuvettes that you may use in the lab. Some cuvettes are clear on all sides, as in this case. Some cuvettes have ridges or frosting on two sides, while the other two sides are clear. It is important to handle the cuvettes carefully to avoid leaving fingerprints and oils from your skin on the surfaces. When working with completely clear cuvettes, only handle the cuvette at the top. When using frosted or ridged cuvettes, make sure you only handle the frosted or ridged sides and avoid the clear side. When you enter the lab, the spectrophotometer will already be on, warmed up, and ready for use. The display should already be in absorbance mode A, as indicated on the display. If it is not, simply press the ATC button until you see the A at the end of the display. In this class, we will only be measuring absorbance, so the display should always be set to absorbance mode. Now you are ready to set the wavelength and blank the machine. As you can see here, the machine is currently set at 450 nanometers. To set the wavelength, use the two buttons labeled NM arrow up or NM arrow down. Make sure you use these buttons and not the arrows near the display area. In this demonstration, I will be setting the wavelength to 430 nanometers by pressing the NM arrow down button. When making changes in wavelengths that are far apart, it is better to press and hold the button rather than pressing and releasing the button 1 nanometer at a time. Now that the wavelength has been set to 430 nanometers, the machine must be blanked before the absorbance of a sample can be read. For your experiment, you will be provided with two cuvettes. One will contain the blank or sample solvent, and the other will be used for your sample. Pick up the cuvette, touching only the top or the ridged sides to avoid fingerprints. It is important to gently wipe the clear parts of the cuvette with a Kim wipe to remove any fingerprints, but not so hard that you scratch the cuvette. Fingerprints and scratches can affect the passage of the light beam through the sample and therefore affect your absorbance reading. Once the blank cuvette has been cleaned, open the lid of the spectrophotometer and insert the blank firmly and carefully in the sample chamber. It is important to keep the clear side of the cuvette facing you to allow the light beam to pass through the cuvette. If you look closely, you will see an arrow beside the slot for the cuvette that shows the pathway of light. Make sure the clear sides of the cuvette are positioned in line with the arrow. Once the cuvette has been fully inserted, close the lid of the chamber. Press the 0 ABS 100% T button to blank the sample. When the machine is done setting the blank, the window will display the wavelength that has been set and 0.000A, or zero absorbance. By setting the blank, we have canceled out all the absorbance due to the solvent used in the experiment. To read the absorbance of a sample, open the lid to the chamber and remove the blank cuvette. Select the sample cuvette and wipe it clean with a Kim wipe. Insert the cuvette, once again making sure the cuvette is positioned with the clear side facing you. Close the lid to the chamber and wait for the reading to stabilize. No adjustments to the machine are necessary. In our case, the absorbance reading is 0.680. Absorbance does not have units. Anytime you change the wavelength, you must re-blank the machine following the steps described to set the wavelength and blank the machine. Begin by removing your sample. Set the wavelength using the NM arrow up or NM arrow down. Using the blank sample, blank the machine to set the absorbance to 0 for the new wavelength. Remove the blank. Insert your sample and record your absorbance reading. Here are some important reminders. Always handle the cuvette carefully to avoid fingerprints by holding either from the top of the cuvette or on the frosted sides. Always wipe the cuvette with a Kim wipe to remove fingerprints, oils, or sample droplets. Always insert the cuvette with the clear side facing you. Always blank the instrument before taking an absorbance reading. Thank you for joining me in this video showing how to use a spectrophotometer in biology lab. Good luck with your experiments.
So any questions about the spectrophotometer? It should look familiar to many of you, right?
Yes.
Okay, so now we're going to watch another video on, let's see, how to calculate CFUs.
Grammarly does more than catch errors. With Grammarly, you can find really good...
So again, it's a number of colonies times the dilution factor. In this case, you divide by the volume that you put on the culture plate, if it's something other than 1 ml. So this is the other way of doing it. The way that I showed you to do it includes the volume you put on the plate in the calculation of the dilution on the plate. So if you do it ahead of time, your dilution factor already included the amount you put on the plate. If you wait to the end, then you divide by the volume that you put on the plate. And again, if it's a 1 ml, this doesn't make any difference, right? Because anything divided by 1 is that same thing. Anything times 1 is still that same thing. It's only if you're using a volume different than 1 ml to put on the plate. So if the plate had a dilution factor of 10 to the 6, and you got 130 colonies on that plate, then 130 times 10 to the 6, put in proper notation, is 1.3 times 10 to the 8 CFUs per milliliter. Right, so is that clear? I mean, this should be dilution factor. So this problem is a little bit more complicated. So they're not using standard amounts to do their dilution. Here they're going to take 5 milliliters of their bacterial culture and they're going to add it to 45 milliliters of sterile broth or sterile water. So you have to calculate your dilution, right, based on that. Then they perform 2 more dilutions, and these are 1 to 100 dilutions. Then they put 0.1 milliliter onto the agar plate. And when they count colonies, they find a plate with 137 colonies on it. So you have to calculate your dilution and dilution factor based on this information. And then give me your CFUs per milliliter. So remember, we're adding 5 mLs to 45, right? So your part divided by the whole. And then they perform 2 more dilutions, which are 1 to 100 dilutions. And then they plate 0.1 milliliters. So I'll give you a few minutes to work on that calculation. And if you think you have the answer, let me know. So when you think you have the answer, let me know. Anybody have anything? Anything? Are you stuck?
So,
Yes?
the total volume is 50, right?
Right, the total volume is 50.
So, I'm confused here because is it going to be part of our whole 5 over 50?
Yep, 5 divided by 50. Now what's 5 divided by 50?
That's 10.
5
over
divided
10.
by 50.
0.1.
0.1, right? And so what is 0.1 exponentially?
0 to the negative of 1.
Right, so 10 to the negative 1.
10 to the negative of 1.
And then we perform 2 dilutions that are 1 to 100 dilutions. Exponentially, what is 1 over 100?
10 to the power of 1.
No, 1 over 100.
0.1.
Right, but exponentially, what is it? 1 over 100 is 10 to the what?
Power of 2. Negative
Yeah,
2.
right, negative 2, so 10 to the negative 2. And that happens twice. And then we're going to spread 0.1 milliliters onto the plate. So exponentially, what is 0.1?
10 to the power of
Well, negative 1.
negative 1.
Right? So 5 over 50 is 10 to the negative 1. And then we have 2 10 to the negative 2 dilutions, and then another 10 to the negative 1. So negative 1, negative 2, negative 2, negative 1. And we get 10 to the what?
Negative 4.
We got 10 to the negative 1, and then 10 to the negative 2, and then 10 to the negative 2, and then 10 to the negative 1 again. So all that together.
10 to the negative 5.
1 plus 2 plus 2 plus 1.
3, 4, 5.
6. Right, 10 to the negative 6.
Plus 1.
Right, so what's the dilution factor of 10 to the negative 6? What's the dilution factor for 10 to the negative 6?
The 10 to the positive of 6.
Right. So now it's 137 times 10 to the 6 put into proper notation. And what would that be?
That's 1.37 multiplied by 10 to the power of 7.
Well, you move the decimal point twice. And so the exponent has to get bigger by 2. Right, so 10 to the 6 becomes 10 to the 8.
8, okay.
So 37 times 10 to the 8. So questions on that? So yeah, we have one more interesting video which shows you technique as well as explaining how to do the dilutions. And in this video, we're also going to see the pour plate technique. So instead of spreading the sample, your diluted sample on the surface of the agar plate, the diluted sample is added to a sterile empty petri dish, and then sterile agar is poured in to mix with the bacteria. So both techniques work. The spread plate's just easier to do in lab, and it's a bit easier to interpret.
The pour plate technique using serial dilutions is one method for quantifying bacteria in a sample. A dilution is created by measuring a volume or weight of a sample and adding it to a volume of sterile water, thus making the resulting solution less concentrated than the original. When we repeat this process for each new tube, thus making a series of more and more dilute samples, it is called a serial dilution. In this illustration, a series of 1 + 9, or 1 in 10 dilutions yields a 10-fold serial dilution. When these serial dilutions are combined with agar and incubated, the microbiologist can then make a count of the resulting colonies, which can be used to estimate the number of viable bacteria in the original sample. We're going to estimate the number of viable bacteria on alfalfa sprouts. Because the quantity of bacteria on these sprouts is unknown, we'll do a series of dilutions to ensure we are able to accurately estimate that quantity. Let's look now at the calculations involved in making serial dilutions. Each dilution blank contains 9 milliliters of sterile distilled water. 1 milliliter of a liquid sample transferred to 9 milliliters gives 1 milliliter of sample in a total volume of 10 milliliters. 1 over 10 can also be written as 0.1, which is equal to 10 to the negative 1. Thus, the dilution of the first tube is 10 to the negative 1. If a solid sample is used instead, 1 gram of sample is considered the equivalent of 1 milliliter of a liquid sample. 1 gram of solid sample transferred to the first 9 milliliter dilution blank is also considered a 10 to the negative 1 dilution. If we transfer 1 milliliter of the 10 to the negative 1 dilution to the second 9 milliliter dilution blank, that transfer is also a 10 to the negative 1 dilution. But the total dilution in the second tube is now the product of the two dilutions, or 10 to the negative 1 multiplied by 10 to the negative 1. Remember, whenever you multiply two terms with the same base, you add the exponents. So the total dilution in tube 2 is 10 to the negative 2. It's important to note that there are alternate methods for performing a serial dilution other than the 1 milliliter in 9 milliliter scheme. One of the most common alternate dilution schemes uses 99 milliliter dilution bottles. In this case, 1 milliliter in 99 milliliters is a 1 to 100 or 10 to the negative 2 dilution. Let's review how to calculate dilutions using our original 1 milliliter in 9 milliliter dilution scheme. For any single dilution, you will divide the amount transferred by the new total volume of that tube, the sum of that amount transferred and the amount already present in the dilution blank. Don't forget, a single dilution is only the dilution you've just made with the current transfer. Calculating a single dilution does not take into account that the source of that transfer may be already diluted. To calculate the total dilution in any one dilution tube, you must multiply the dilution of the transfer, the single dilution, by the dilution of the previous dilution tube, the tube from which the transferred amount was taken. Now that we know how to calculate dilutions, let's use our serial dilution method to estimate the number of bacteria on sprouts. First, label each blank with the total dilution it would have. Aseptically weigh 1 gram of sprouts. Using aseptic technique, remove the cap from the first 9 milliliter dilution blank, flame the mouth, then use forceps to transfer sprouts to the blank. Flame and recap the tube. This is the 10 to the negative 1 dilution, since we're adding 1 gram of sprouts to 9 milliliters of sterile water. Recall, if using a liquid sample, such as water or milk, we would simply transfer 1 milliliter of sample to the first 9 milliliter dilution blank to create this 10 to the negative 1 dilution. Hold the tube at an angle and mix the contents, either with a vortex mixer or by flicking the tube vigorously with your index finger. Use a sterile pipette to make each subsequent transfer. Open the pipette wrapper at the top and attach the aspirator while holding the pipette by its wrapper. Do not touch the pipette with your hands. Using aseptic technique, transfer 1 milliliter from the 10 to the negative 1 dilution to the 10 to the negative 2 dilution blank. Mix the contents well. This 10 to the negative 2 dilution can now be used to create the next dilution. Being careful to avoid contamination, insert a new pipette into the aspirator. Then, pick up the 10 to the negative 2 dilution and aseptically withdraw 1 milliliter. Remove 1 milliliter from the 10 to the negative 2 dilution and transfer to the next 9 milliliter dilution blank. You have now created a 10 to the negative 3 dilution. Then, make the remaining transfers in the dilution scheme. If your dilution scheme requires you to use 99 milliliter dilution bottles, you will mix the contents by shaking the bottle 20 times through a 35 centimeter arc. Once all of the dilutions have been made, transfer inoculum to empty sterile petri dishes. In this example, we will plate the last 4 dilutions. The melted trypticase soy agar should be kept in a warm water bath between 45 and 50 degrees Celsius. If the agar is too hot, it will kill the bacteria. If the agar is too cold, it will solidify in the vessel or as it is poured into the cold petri dishes. Wipe the outside of the vessel with a paper towel, being careful not to contaminate the mouth. Gently swirl the nutrient agar, taking care not to make bubbles. Remove the plug with the pinky and ring finger of the hand that lifts the plate lid. Flame the mouth of the vessel. Use your thumb and forefinger to remove the lid from the plate. Pour 12 to 15 milliliters of the melted agar into the plate so that it is about 1/3 full. Immediately replace the lid. Swirl the plate gently. Repeat this procedure for all plates. After all plates have been poured, flame and re-plug the flask. Set aside. Gently swirl the poured plates for about 30 seconds. Don't let the agar splash onto the lid or over the edge. Then, leave the plates to cool so the agar can solidify. The solidified agar is more opaque than the liquid. After the agar has solidified, invert plates and incubate at 35 to 37 degrees Celsius for at least 24 hours. Plate counts assume that each live bacterium grows to produce a single colony. In reality, some cells remain clumped and can also form a single colony. Therefore, results are reported as colony-forming units, or CFUs, per milliliter or gram of a sample, instead of cells per milliliter or gram. To obtain a statistically reliable estimate of the quantity of bacteria in your sample, select the plate that contains between 30 and 300 colonies. In this set of plates, that would be the third plate. Plates with too many colonies to count are labeled TNTC, which stands for too numerous to count. Plates that have fewer than 30 colonies are labeled TFTC, which stands for too few to count. Not all colonies will be on the surface, because some bacteria were trapped in the agar as it solidified. These colonies are noticeably smaller and elliptical. Even these embedded colonies must be counted. To calculate the number of bacteria per gram or per milliliter of the original sample, use this formula. The number of colonies on the countable plate is multiplied by the reciprocal of the dilution of the plate counted. In our thus making a series of more and more dilute samples, it is called as fewer than 30 colonies are labeled TFTC, which stands for too few to count. Not all colonies will be on the surface, because some bacteria were trapped in the agar as it solidified. These colonies are noticeably smaller and elliptical. Even these embedded colonies must be counted. To calculate the number of bacteria per gram or per milliliter of the original sample, use this formula. The number of colonies on the countable plate is multiplied by the reciprocal of the dilution of the plate.
So when they did their experiment here, they used 1 milliliter for everything, including how much they put on the plates. And so that's why they're not bothering to tell you to divide by 1 milliliter at the end or do anything like that, because they use 1 ml for everything. And those colonies that are embedded in the agar, that does make it a little bit more difficult to count. And so overall, this technique is harder to perform, which is why we prefer to spread our sample on the surface.
Counted. In our example, there were 89 colonies on the plate, with a dilution of 10 to the negative 5. We multiply the 89 colonies by the reciprocal of that dilution, 10 to the 5th, to achieve a result of 89 times 10 to the 5th CFU per gram. Simplified to two significant figures, the concentration is expressed as 8.9 times 10 to the 6th CFU per gram. After recording your data, properly discard your plates for autoclaving.
And so the reason why they used 1 gram is because, as you recall, 1 gram is equal to 1 milliliter. And so that's why they could use 1 gram instead of 1 milliliter. It was an interesting experiment what they were doing. But any questions? I know you have questions, but any that you want to ask? Or you need to think about everything for a while? Is everybody still awake? Yes, you're still awake, Sarah? So